Welcome back to the deep dive. Our blueprint for human life, it's built on these 46 chromosomes, usually perfect, but, well, what happens when that blueprint has errors? Today, we're tackling a really complex area, chromosomal disorders. We're going to try and cut through the jargon, use our expert sources, and give you the essential facts.
And it's a huge area. I mean, these disorders, major changes in number or structure of chromosomes, they're behind a lot of early pregnancy loss. The estimates are something like 7.5% of all conceptions.
Yeah.
Well, it's a pretty high error rate, fundamentally.
Wow, 7.5%. That's staggering.
It is, though thankfully only about 0.5% to maybe 1.0% of actual live births end up showing these major problems.
So to even start understanding this, how do scientists actually see these problems? I know the key tool is something called karyotyping.
That's right, karyotyping. It's quite a clever technique, really. They take cells that are dividing, often blood cells, lymphocytes, and use chemicals like calcimate to stop them right in the middle of division, metaphase.
Metaphase?
Yeah.
That's when the chromosomes are all condensed and visible.
Exactly. That's the only time you can really see them individually. Then they stain them, usually gene sustain, which gives you G-banding.
G-banding, and that creates a pattern.
A unique pattern, yes, like a barcode for each chromosome pair. Distinct light and dark bands, absolutely crucial for identification.
And the detail matters, I assume?
Oh, immensely. Standard banding gives you maybe 400 to 800 bands per set. But if they catch the cells a bit earlier, in prophase, the chromosomes are longer. And you can get up to 1,500 bands, much higher resolution. That lets you spot smaller, more subtle structural changes you'd otherwise miss.
Okay, so they get this picture, this map, and then there's that shorthand notation you see, like for Down syndrome, 47XY plus 21. How does that work?
It's like a precise address. First number is the total count of chromosomes 47 here, instead of the usual 46. Then the sex chromosomes XY for male. And finally, the specific issue. Plus 21 means an extra copy of chromosome 21.
Simple enough, and for bits within a chromosome.
We use P for the short arm, think petit, and Q for the long arm. So something like XP21.2 tells you exactly where to look. X chromosome, short arm, region 2, band 1, subband 2, a very specific location.
Got it. Okay, let's unpack the main types of errors, starting with the most common, which is just having the wrong number of chromosomes, aneuploidy.
Right, aneuploidy. And the main reason for that, overwhelmingly, is nondisjunction.
Nondisjunction sounds like things not splitting apart correctly.
That's basically it. During meiosis, when eggs and sperm are made, either the paired chromosomes or the sister chromatids fail to separate. So you end up with a gamete, an egg, or sperm that has one too many chromosomes, that's N plus 1, or one too few, N1.
And when that gamete gets fertilized,
You get a zygote that's either trisomic, 3 copies of a chromosome like 2N plus 1, or monosomic with only 1 copy, 2N1.
trisomy 21 is the classic example. But what about monosomy? Losing a whole autosome, a non-sex chromosome, can that ever happen in a live birth?
Almost never. For autosomes, losing one entire copy is typically lethal very, very early on. It just highlights how sensitive our bodies are to gene dosage. We really need those 2 copies of almost every autosomal gene to develop properly. Too much or too little information is usually catastrophic.
Okay, so that's errors in the egg or sperm, but then there's mosaicism. That's different, isn't it? It happens later.
Correct. Mosaicism isn't from the gamete. It's an error in mitosis after fertilization, early in the embryo's development. So the individual ends up with 2 or more different cell lines. Some cells might be normal, say 46XY, while others have the abnormality, maybe 47XY plus 21.
Wow, so like a patchwork person, chromosomally speaking.
In a sense, yes. And the clinical outcome, how severe it is, really depends on which tissues have the abnormal cells and what proportion of cells are affected. Mosaicism involving the sex chromosomes is actually relatively common. Autosomal mosaicism is quite a bit rarer.
Fascinating. Okay, so that covers number errors. But what about when the count is right, 46, but the chromosomes themselves are broken or rearranged, structural errors?
Right. This happens when chromosomes break and then rejoin incorrectly. There are several main types. A deletion is pretty straightforward. You just lose a piece of a chromosome. It can be in the middle, interstitial, or at the end, terminal.
An inversion?
That's when a fragment breaks off, flips 180 degrees, and then reinserts itself back into the same chromosome. Carriers are often phenotypically normal because they haven't lost any genetic material, but they could have problems making balanced gametes later on. Big implications of reproduction.
And then an isochromosome. That sounds unusual.
It is. That's when a chromosome divides the wrong way. across the centromere. You lose one entire arm, and the other arm gets duplicated. So you end up with a chromosome made of two identical arms, either two short arms or two long arms. The most common one seen in live births involves the X chromosome's long arm, written IXQ10.
So you're missing all the genes on one arm and have triple the genes on the other. A dosage problem again, but caused structurally.
Precisely. It's a structural change leading directly to a gene dosage imbalance.
Okay, and finally, translocations. Moving pieces between different chromosomes. There are two key types here.
Yes. First is a balanced reciprocal translocation. This is where two different chromosomes basically swap segments. The person carrying this usually looks and functions completely normally because overall they still have the right amount of genetic material, it's just rearranged.
But the problems come later.
Exactly. When they produce eggs or sperm, it's very difficult to segregate those rearranged chromosomes correctly. They have a high risk of producing unbalanced gametes, which can lead to miscarriages or children with significant genetic disorders.
And the second type, the one linked to Down syndrome.
That's the Robertsonian translocation, or centric fusion. It specifically involves two acrocentric chromosomes. Those are the ones where the centromere is very close to one end, like chromosomes 13, 14, 15, 21, and 22. These two acrocentric chromosomes fuse together near their centromeres. A tiny fragment containing mostly redundant genetic material is usually lost in the process. The carrier ends up with only 45 chromosomes, but is typically healthy because essentially all the important genetic info is still there. But if chromosome 21 is involved in that fusion.
Ah, that's the link. That explains the familial form of Down syndrome.
Precisely. About 4% of Down syndrome cases are due to this Robertsonian translocation, often inherited from a carrier parent.
Okay, let's focus on the outcomes. Trisomy 21, Down syndrome. It's the most common chromosomal disorder, right?
By far. About 1 in 700 live births. And 95% of the time is caused by that nondisjunction. We talked about getting a whole extra chromosome 21. And interestingly, in about 95% of those nondisjunction cases, the error happened in the formation of the egg, not the sperm.
And the link with maternal age is very strong.
Extremely strong. The risk goes from about 1 in 1,500 for mothers under 20 up to, well, a shocking 1 in 25 for mothers over 45. It's a dramatic increase.
The clinical features are well known, the intellectual disability, the facial characteristics.
Yes, severe intellectual disability is the leading genetic cause. Characteristic features like a flatter facial profile, upward slanting eyes, epicanthic folds, but also major internal issues. About 40% have congenital heart defects, often atrioventricular septal defects. That's a major source of problems.
And the cancer risk increase is just astounding.
It really is. A 20-fold higher risk of a type of leukemia called BAL, and a 500-fold increased risk for another type, acute myeloid leukemia. It's remarkable.
And there's a connection to Alzheimer's disease, too. How does that work?
It comes back to dosage. The gene for amyloid precursor protein, APP, is located on chromosome 21. Having 3 copies of that gene means individuals with Down syndrome overproduce amyloid beta protein throughout their lives. As a result, virtually all of them develop the brain pathology of Alzheimer's disease by the time they're 40 or 50.
Just shows how critical having the right amount of even one protein can be.
Absolutely. And to really hammer home the dosage point, we should briefly mention trisomies 18 and 13 Edwards and Patel syndromes.
Right, they're much rarer and much more severe.
Far more severe. They involve chromosomes carrying even more crucial genes, perhaps. The level of malformation is profound. Sadly, most infants born with trisomy 18 or 13 don't survive more than a few weeks or months. It really underscores that while an extra chromosome 21 is serious, extra copies of most other autosomes are simply incompatible with sustained life.
Okay, shifting gears slightly, what about smaller errors? Not a whole chromosome, but a missing piece, like in 22q11.2 deletion syndrome.
Excellent example. This is a microdeletion. You're missing a small segment, about 1.5 million DNA base pairs, from the long arm of chromosome 22. It causes a huge spectrum of issues, sometimes called DiGeorge syndrome if the thymus gland is badly affected, leading to T-cell deficiency, or velocardiofacial syndrome, focusing on heart defects and palate issues.
And distinct facial features are common, too.
Yes, and heart defects are very common. But what's really striking is the neuropsychiatric risk. About 25%, a quarter of adults with this deletion, develop serious psychotic illnesses, particularly schizophrenia. It's a fascinating link between a small genetic deletion and complex mental health conditions.
Okay, let's move to the sex chromosomes. We said autosomal imbalances are usually devastating, but having an extra X or Y or missing an X seems better tolerated. Why is that?
It all comes down to a process called X inactivation, sometimes called the lion hypothesis, after Mary Lyon, who proposed it. Basically, very early in the development of a female embryo, XX, one of the two X chromosomes in each cell gets randomly shut down, inactivated. It condenses into a little structure called a bar body.
So females effectively only use one X chromosome in most cells, just like males, XY.
Pretty much. It's a dosage compensation mechanism. It ensures that males and females have roughly the same dose of proteins produced by X-linked genes. So females are actually mosaics. Some cells use the paternal X, some use the maternal X.
But you said roughly the same dose. It's not perfect.
Not entirely. It turns out about 30% of the genes on the short arm, XP, and a few on the long arm actually escape inactivation. They remain active on both X chromosomes in females, and this is crucial. It explains why missing an X chromosome entirely, as in Turner syndrome, 45X, still causes significant problems because you lose the contribution of those escaping genes.
And the Y chromosome's main job?
Its main job is sex determination. Thanks to the SRY gene sex-determining region Y, presence of SRY triggers male development. Simple as that, fundamentally.
Okay, let's look at the two main sex chromosome disorders. First, Klinefelter syndrome, 47,000XY.
Right. This is the most common cause of male hypogonadism, basically underdeveloped tests. The phenotype often includes being tall, particularly long legs, a more unicoid body shape, small tests, and sometimes gynecomastia, which is breast development.
And the cause is the extra X.
Yes. Specifically, the overexpression of those genes that escape X inactivation. For example, the SHOX gene involved in height is on the X chromosome and escapes inactivation. Having two active copies contributes to the tall stature seen in Klinefelter. There are also issues with how the body responds to androgens.
And the flip side, Turner syndrome, 45X. Only one X chromosome.
This is female hypogonadism. The defining features are short stature again, linked to having only one copy of that SHOX gene, haploinsufficiency, and ovarian failure. Phenotypically, you might see things like a webbed neck, a broad chest, and, importantly, cardiovascular problems. Coarctation of the aorta, a narrowing, is particularly common and a major health risk.
And the ovarian failure.
You need two X chromosomes for ovaries to develop and function properly. With only one X, the oocytes, the egg cells, are lost much faster than normal. It leads to what's sometimes dramatically called menopause before menarche. The ovaries become streaks of fibrous tissue.
Okay, one last major category, shifting from these large structural or numerical changes to something more molecular. Fragile X syndrome, FXS.
Right. FXS is the second most common inherited cause of intellectual disability, after Down syndrome. It used to be diagnosed by seeing a fragile site on the X chromosome under specific lab conditions.
But now we know the actual mechanism is different.
Yes, we know it's caused by an expansion of a specific DNA sequence, a CGG trinucleotide repeat within the FMR1 gene, located on the long arm of the X chromosome, X227.3.
And the number of these CGG repeats is absolutely critical.
Totally critical. A normal person has somewhere between 6 and maybe 55 repeats. Then there's a gray zone called the premutation, typically 55 to 200 repeats. But when the number of repeats expands beyond about 200, usually over 230, it becomes a full mutation.
And that full mutation causes the main Fragile X syndrome.
Exactly. That massive expansion triggers a chemical change called methylation, which effectively silences the FMR1 gene entirely. You lose the FMRP protein. And FMRP is vital for normal brain development, particularly for regulating protein synthesis at synapses, impacting learning and memory. Loss of FMRP causes the intellectual disability and other features of FXS.
This leads to those weird inheritance patterns, like anticipation and normal transmitting males. How does that work?
It's fascinating. The premutation is unstable. A male can carry the premutation, a normal transmitting male, and be largely unaffected himself. But when he passes the X chromosome to his daughters, they inherit the premutation. But here's the key. The repeat sequence is particularly unstable and prone to further expansion when it passes through female meiosis during egg formation.
So the daughter might expand her premutation into a full mutation when she has children.
Precisely. So the risk of having an affected child and the severity tends to increase in successive generations. That's anticipation. The premutation itself is like a ticking time bomb, genetically speaking.
But the premutation itself isn't harmless, right? It causes different problems.
That's a really important point and a different mechanism. While the full mutation causes FXS through loss of FMRP function, the premutation causes problems through a toxic gain of function. In premutation carriers, the FMR1 gene is still transcribed into mRNA, but the mRNA molecule itself, with its long CGG repeat track, is abnormal. It seems to gum up the works in the cell by binding to and sequestering important RNA-binding proteins. This leads to distinct disorders like Fragile X-associated tremor ataxia syndrome, FXTAS, in older carriers, and premature ovarian failure in female carriers. Different mechanism, different disease.
Amazing complexity. So we started with G-banding. Given all this microdeletions, repeat expansions, how are these conditions actually diagnosed today?
Well, G-banding is still useful for large changes, but its resolution is limited, millions of base pairs. For smaller things, we need better tools. FHH fluorescence in situ hybridization is key. It uses fluorescent DNA probes that bind to specific chromosome regions. It can detect much smaller deletions or duplications down to kilobases. Essential for confirming things like 22q11.2 deletion that standard karyotyping might miss.
And prenatal testing has advanced too, hasn't it?
Hugely. NIPS, non-invasive prenatal screening, is a major development. It analyzes fragments of cell-free fetal DNA that circulate in the mother's blood. By measuring the amount of DNA from, say, chromosome 21, they can screen for trisomy 21 with very high accuracy, non-invasively. Though often, an invasive test like amniocentesis for conventional karyotyping is still needed for a definitive diagnosis.
And for things like Fragile X.
That's now diagnosed directly using PCR-based methods to precisely measure the number of CGG repeats. Much more accurate and reliable than looking for the old cytogenetic fragile site. Technology has really moved on.
So this deep dive really shows the incredible range of things that can go wrong with our chromosomes. Big number changes, tiny deletions, even these stuttering DNA repeats. But it seems the core issue, again and again, comes down to gene dosage having too much or too little of critical genes, like APP in Down syndrome or SHOX in Turner and Klinefelter.
That's spot on. What's really fascinating, I think, is that the size of the error doesn't always predict the severity. A tiny change affecting a crucial gene, or one that bypasses our compensation mechanisms like X inactivation, can be devastating. It really highlights the exquisite balance required for normal development and maybe how much more we still need to learn about managing that balance.
A really profound point to end on. It certainly changes how you think about genetic vulnerability and the whole process of development. Thank you for walking us through that complex blueprint today.